Yung nakubukin.
Kana guys, nag-echo pa ko, wala lang.
Wala na.
Wala na.
Siya.
Sige, sige.
Sige, go.
Okay, start ka. Okay. Maingay ka talaga. Tago ka muna sa kwarto mo. Okay. Good morning. Our first group will discuss etiology, pathogenesis, structural causes, and virus known as human papilloma relatively small and lacks an envelopment. Oh shoot. Balik. Sorry. Good morning. Our first group will discuss etiology, pathogenesis, structural causes, and symptoms of human papilloma virus. The DNA virus known as human papilloma virus or HPV, this virus is relatively small and lacks an envelope that can alter cellular structure and cause the development of abnormal tissue. It is highly contagious, a common virus that can affect different parts of your body, and it is sexually transmitted infection. This has persisted for a long time, can result to cancer, especially cervical cancer and warts.
Now, let's know about the etiology of human papilloma virus. And we have two main possible causes. First is the skin to skin contact, which is the primary cause of this virus. A person may get infected by simply skin to skin contact, most especially during sex intercourse. It is most often disseminated through open wounds, skin abrasions, and minor tears, but it is also capable of being passed from person to person via intimate skin to skin contact. Second is the direct contact. In direct contact, people who are at high risk of getting infected are usually those who engage of having multiple sexual partners because by this means the spread of this viral infection speeds up. Other possible cause could be the early age at first intercourse. So note that the human papilloma virus is not passed on by biological fluids like saliva or sperm. Rather, it is transmitted from one individual to another by direct skin to skin contact.
Next slide, please. So, an established omnicavirus is the human papillomavirus known as HPV. Papillomavirus is a small virus, or I can call it a tiny danger, with a simple capsid surrounding a circular DNA genome. The caspid PBD entry 3J6R includes 360 copies of the major caspid chain called L1. A second caspid chain called L2 is found on the inside and may help to transmit the virus. with the packaging the genome virus instead, like a simian virus 40, the L1 chain forms 72 pentameric capsomers, which then interact with one another through long, flexible tails.
There is a specific type or number for each human papilloma virus. High risk HPV strains include 16, 18, 31, 33, 35, 39, 45, 51, 52, 56, 58, 59, 66, and 68. The bulk of HPV related malignancies are caused by two of these, HPV 16 and HPV 18. An example of a flat, thin cell type that contains HPV is the epithelial cell. On the surface of the skin, they are visible. Additionally, they are present on the surfaces of the cervix, anus, vulvar, vagina, and penis. They may also be found in the mouth and throat. Next slide, please. Next slide again, please. So, viral components and physical properties. Papilloma viruses are icosahedral DNA viruses that are tiny, non-enveloped. This implies that there is no protective envelope around the virus, which means it can exist. It has also a measure of 52 to 55 nm in diameter. The virus is made up of 72 pentameric capsomeres wrapped around single double-stranded DNA molecule with an approximate length of 8,000 base pairs attached to cellular histones. A protein-infused capsid surrounds the DNA that makes up HPV. The virus genetic makeup serves as its template, while the capsid acts to enclose and safeguard it. The genetic material is transported to virus host cells by the capsid, which functions as a vehicle. Next slide, please. Two structural proteins in the capsid. First is the late L1, 55 kDa in size, 80% of total viral protein. And next is the L2, 70 kDa in size. Both the L1 and L2 proteins are virally encoded. In mammalian or non-mammalian expression systems, the expression of L1 alone or in conjunction with L2 can result in the production of virus-like particles. The intact virion has a sedimentation coefficient of 300 and a density in cesium chloride of 1.34 gml. Next slide, please. So this is the HPV genome. So 8 ORFs, which are all transcribed from a single DNA strand molecule with a genome size of roughly 800 BP, are found in the genomes of all HPV strains. Functional ORF components. So first one is the early E section, which codes for the E1 to E7 proteins required for viral replication. Next is the late L region, which codes for the L1 to L2 structural proteins needed for virion assembly. Next is the long control genome. The term long control region, rather, refers to a substantial non-coding region that comprises CIS regions required for viral DNA replication and transcription. The early promoter, such as P97 and HPV31, is where the viral E proteins are transcribed, whereas the late promoter is where the viral L proteins are primarily transcribed. The papilloma viruses varies in size and number of ORFs, but they all have well-conserved core genes for replication, such as E1 and E2, and packing, such as L1 and L2, as well as very diverse genes for cell entry, immunological infiltration, and virus release, such as E6, E7, E5, and E4.
So this is the causing cancer. The papillomavirus genome encodes two tiny proteins with the names of E6 and E7 that in highly harmful strains function as a cancer-causing oncogenes. Together, they encourage cells' abnormal development and suppress their built-in defenses. They affect a wide range of cellular signaling proteins, which regulate various processes like death and proliferation. E6 is shown performing one of its horrible chores in PDB entry 4XR8. The PB53 tumor suppressor and ubiquitin ligase are building linked in instruction which will ultimately result in the proteasomes. Next, destruction of PB3. The virus prevents the cell from employing adaptopsis to end the infection this way, which may result in cancer. So in this slide, which is shown now, it is the heparin molecules on the surface of the infected cells are bound by papillomavirus heparin chains. On the left, in green, are recognized by lysine line groups on the surface of the virus according to crystallographic structure of the isolated L1 capsomers, PBDN3 5W10. Antibodies against similar L1 capsomer struggles on the right, as you can see in the slide, there's blue, demonstrate their ability to prevent this identification preventing attachment to the cell. This concludes the section of the structures of organisms. Mara Group will now cover the pathophysiology on the next following slide.
I will now proceed to the pathophysiology of the human papilloma virus. As mentioned previously, papilloma viruses are non-enveloped double-stranded viruses with icosahedral symmetry. They are also very specific and cannot infect different species other than humans. And although some overlap exists, most papilloma viruses have distinct anatomic predilections, so they only infect certain epidermal sites such as skin or genital mucosa. This means that an HPV infection in one area of the body shouldn't spread to another since it doesn't reach the bloodstream and it is more of a skin associated virus. Specifically, the first thing that happens is that the basal keratinocyte of the epidermis are infected by HPV, and this is presumably through disruptions of the skin or mucosal surface. And it is also important to take note that at this stage, the virus remains latent in the cell as a circular episome in low copy numbers. So this means that the virus is already existing but it is still inactive. However, as the epidermal cells migrate to the surface and differentiate, the virus is triggered to replicate and mature. Papilloma viruses are thought to have two modes of replication. The first one is stable replication of the episomal genome in the basal cells, and the second one is runaway or vegetative replication in more differentiated cells. And after all of that, the virus is now present in high copy numbers and is shed in the exfoliation cells. And this process of virus replications alter the character of the epidermis. And although all cells of a lesion contain the viral genome, the expression of viral genes is tightly linked to the state of cellular differentiation. So, as I've said earlier, most viral genes are not activated until the infected keratinocyte leaves the basal layer, and production of virus particles can only occur in highly differentiated keratinocyte.
Now, numerous viral genotypes can change cells and are associated with epidermal carcinoma. In benign or low risk HPV lesions, such as those connected to HPV type 6 and 11, the HPG genome resides as a circular episomal DNA separate from the host cell nucleus. In malignant lesions, or the genomes of high risk HPV type 16 and 18, are generally incorporated into the DNA of the host cell. A distinguishing characteristic of malignant transformation is thought to be integration of the viral genome into the genome of the host cell. Additionally, the tumor suppressor proteins P53 and RB of the host have been demonstrated to be inactivated by high risk HPV proteins E6 and E7, which cause unchecked host cell proliferation and malignant transformation. As a result, human papillomavirus leads to the emergence of various wart types, such as skin warts, plantar warts, flat warts, anogenital warts, laryngeal papillomas, and several cancers, such as those of the cervix, vulva, penis, and anus, as well as a subset of head and neck cancers, all occur at low temperatures without a host. Therefore, an individual with plantar warts can spread the virus by walking barefoot.
Now let's proceed to the clinical manifestations of human papillomavirus. As we know, clinical manifestations are the signs and symptoms. The signs of human papillomavirus include recurrent respiratory papillomatosis, cervical cancer precursors or HSIL, and cancers such as cervical, anal, vaginal, vulva, penile, and oropharyngeal. Next slide, please. First is the recurrent respiratory papillomatosis. This is a very rare condition caused by viral wart development in the larynx. Less often, the disorder affects the area within the oral cavity, trachea, and bronchi. Only in rare cases do these growths spread to affect the lungs. Next slide, please. Next is the cervical cancer precursors. It is a squamous cell abnormality caused by the human papillomavirus. Although not all HSIL will advance to cancer, it is still regarded as a precancerous lesion and is therefore generally handled assertively. Lastly is the cancer. The first type is the cervical cancer. It is a specific type of cancer that appears in the cells of the cervix, which is the lowest portion of the uterus that joins the vagina. This cancer can affect the deeper tissues of the cervix and may spread to other parts of the body, often the lungs, liver, bladder, vagina, and rectum. Next is the anal cancer, in which cancerous cells form in the anus tissues. Signs of anal cancer include bleeding from the anus or rectum or a lump near the anus. Vaginal cancer is also included. It is a type of cancer in which the muscular tube that links the uterus to the external genitalia develops malignant cells. This type of cancer affects women and more common in women over the age of 60. Next is the vulval cancer. It is a type of cancer that forms on the female genitalia's outer surface. Vulva is the area of skin that surrounds the urethra and vagina, including the clitoris and labia, and it commonly forms as a lump or sore on the vulva that often causes itching. It also includes penile cancer. It is an uncommon form of cancer that results in uncontrolled cell growth on or in a man's penis. It mostly affects the skin of the penis and the foreskin, which is the skin covering the head of the penis. Finally, the oropharyngeal cancer. It is a type of cancer in which cancer cells develop in the oropharyngeal tissue. The signs and symptoms of oropharyngeal cancer include a lump in the neck and a sore throat.
Next is the symptoms of human papillomavirus. So, symptom is a manifestation of disease apparent to the patient himself or the subjective evidence of disease. In most HPV infections, patients are asymptomatic, showing in few to no symptoms. A transient infection is an HPV infection that transpires and then clears. This type of HPV infection appears most typically in younger sexually active women. Next slide, please. Less frequent symptoms include genital lumps and bumps. Since HPV infection causes warts, these warts form in and around the rectum. Genital lumps and bumps also include penile genital warts that are usually 2 to 5 millimeter in diameter and often occur in groups. It is typically present as asymptomatic papules or plaques. Lesions may be filiform, exophytic, papillomatous, verrucous, hyperkeratotic, cerebiform, fungating, or cauliflower-like. Next slide, please. Genital lumps and bumps can also be seen as warts of the vulva and vagina. These are soft, whitish papules with many fine finger like projections or that are sessile. These lesions are commonly found in the introitus and labia and are sexually transmitted infections. However, many people never experience any symptoms. The third one for the genital lumps and bumps is the anogenital warts, are the elevated development of the skin surrounding the anus. They are usually asymptomatic. Epidermoid cyst is a small non-cancerous lumps beneath the skin. UV exposure and HPV viral infection will foster the formation of epidermoid inclusion cysts. They can appear anywhere on the skin, but are most common on the face, neck, and trunk. They are slowly growing and often painless, so they rarely cause problems or need treatment. Genital itching. These can develop in or near the vulva, vagina, or cervix. Warts can appear alone or in cauliflower clusters. Genital warts can cause genital itching, burning, sensation, and tenderness. HPV can also result in common warts, flat warts, and plantar warts. For common warts, these appear rough, raised lumps that form on the hands, fingers, and elbows. For flat warts, these are flat topped, slightly elevated lesions that are darker than the surrounding skin and are most commonly found on the face or neck. Lastly, the plantar warts are hard, grainy growths that typically appear on the foot, most commonly on the heels or balls of the feet.
Here we go. Let's talk about histological examination of human papilloma virus. HPV are considered to have a basaloid and warty subtype of HPV, and this affects younger women. Opposingly, the HPV independent lesions generally have keratinizing morphology and are found commonly in elderly women. The HPV test checks cells for infections with high risk HPV types. The POP test, also called as pap smear or cervical cytology, which collects cervical cells, and this looks for changes caused by HPV. And that if this is left untreated, this turns into cervical cancer. It can also detect cervical cancer cells. The figure shows basaloid tumors in the squamous cells. Basaloid tumors, which can be seen in the mature squamous cells that are characterized for being made up of tiny undifferential cells in which there is a presence of no to little keratinization and koilocytosis. Basaloid component consisting of a solid and lobular growth of small crowded cells with a scant cytoplasm. Next slide.
Now, for figure 2, or the warty type or warty subtype of HPV. For figure 2.1 on the histologic examination, the figure shows a warty subtype of HPV in which it can be defined on having condylamitous features and also pleomorphism, in which pleomorphism are the existence of irregular and variant forms in the same species of microorganisms. Also, there are individually keratinized cells and choliocytic-like cells like pernicular cavitation in which it can be developed by nucleation, nuclear enlargement, and hyperchromasia. While on figure 2.2, the figure shows a warty squamous cell seronoma in which it shows an irregular vericome or verrucum, or in which verrucus has been applied for lesions showing a kerotic exophytic surface composed of sharp or blunt epithelial projections in which keratin-filled invaginations or plugging, but without obvious fibrovascular cores, in which architecture that because of the presence of irregular vermiform architecture has now conspicuous vibrocascular cores. So this is our references on our reporting for today. So this is the group one. Uh, that would be for our discussion about the human papilloma virus. So thank you for listening and good day to everyone.